Hello, my name is Scott Requadt, CEO of Talaris Therapeutics, and I'm delighted to present the Talaris story at Meeting on the Mesa. This is the standard disclaimer regarding forward-looking statements. At Talaris, we have set ourselves an audacious goal. With our proprietary allogeneic stem cell-based therapy, our goal is to potentially profoundly impact three areas of great unmet need relating to immune tolerance. The first of those is organ transplant, where our goal is to enable an individual receiving an organ transplant to have their immune system reprogrammed so as to recognize the donated organ as self without the need for lifelong chronic immunosuppression. We also believe that the same therapeutic administered in substantially the same way can also enable somebody suffering from severe autoimmune disease to have tolerance restored to self. And finally, we believe that our therapy has the potential to be a more attractive alternative to traditional allogeneic stem cell transplant in addressing diseases where you have diseased immune or blood cells, and where the goal normally would be to replace those through allogeneic stem cell-based transplant, but where we believe that our approach could enable that to happen without the need for myeloablative conditioning and potentially across HLA mismatched donors. Our lead therapy, which we call FCR001, is currently in a phase 3 registration study in living donor kidney transplant patients, where our goal is to demonstrate the potential of our therapy to induce durable immune tolerance in these patients to their donated organ. On the basis of compelling phase 2 data, we've received orphan and RMAT designation from the FDA, and we now have a phase 3 registration study underway, the design of which effectively mirrors our optimized phase 2 design. Over the course of the phase 2, we identified a highly predictive marker, which we believe can guide us in predicting which patients are likely to benefit from being able to be removed from immunosuppression and for that immunosuppression removal to be durable. We intend to provide the first clinical update on our open label phase 3 study later this year in November. We also intend to start 2 more phase 2 studies before the end of this year to begin to demonstrate the extensibility of this approach, both across other areas of organ transplant as well as autoimmune disease. And we intend to announce a third indication for our therapy also before the end of the year. We do all of our cell processing and manufacturing in-house in our own facility using a robust and reproducible method. And we're well financed with a $265 million cash balance as at the end of this past June. Our product and the basis of our therapeutic approach is FCR001. It comprises of 3 cell types, all of which are derived from a healthy donor. In the case of living donor kidney transplant, that donor is the donor of the kidney. The most critical of these ingredients is the hematopoietic stem cell. But there are 2 other critical ingredients which we also look to obtain from the donor. One of these is called a facilitating cell, which is a unique non-stem cell type of cell, which is CD8 positive, T cell receptor negative, which our founder has identified as having unique properties to both promote engraftment of stem cells in unmatched recipients, and also to upregulate a number of tolerogenic pathways, which we believe enable the presence of the cell to help obviate or mitigate against the risk of graft versus host disease, even in unmatched donor-recipient pairs. We also identified a specific amount of alpha beta T cells from the donor, which need to be included in our therapy. We administer this therapy with non-myeloablative conditioning, which means we're not wiping out the immune system of the recipient, at doses and protocols optimized over 2 decades of study, and we have issued composition of matter patents protecting this therapy, as well as a large number of trade secrets relating to our manufacture of this product. Our pipeline is as diverse as you would suggest from the initial slide. Our lead indication is in living donor kidney transplant, where, as I mentioned, we have a phase 3 registration study, which we call Freedom 1, now underway. And we'll be starting another study in patients who previously received a living donor kidney transplant within the past 3 to 12 months before the end of this year. We're also going to be starting before the end of this year our first study in autoimmune disease, namely in scleroderma. And we have preclinical efforts to show the extensibility of our approach to also apply to the deceased donor organ transplant space, which is a 4 times larger commercial opportunity. And as I mentioned, we'll be announcing an indication in this last area, the replace indication, before the end of this year. So as many of you know, an individual who's receiving an organ transplant needs to be on immunosuppression for the rest of their life, or they will reject their organ. Unfortunately, these agents are associated with toxicity to the kidney, resulting in kidney function decline over time and the need for retransplant. They're also associated with increased risks of cancer, cardiovascular comorbidities, infection, compliance issues, quality of life, and CNS issues. And our goal is to enable somebody receiving an organ transplant to be relieved of the burden of lifelong immunosuppression. These are our top-line phase 2 data. In our phase 2 study, we were able to demonstrate that 70% of the patients who received our therapy, as well as a kidney transplant, were able to be weaned off all immunosuppression, typically within a year of their transplant. And once we optimized aspects of our phase 2 study, 14 of the last 17 subjects in our study were able to discontinue all immunosuppression. Notably, every single patient who came off immunosuppression has stayed off immunosuppression for the duration of follow-up. And we've now followed the phase 2 cohort for a median of over 6 years, and the longest of these out beyond 11 years with 100% durability. Also, 10 of the subjects in our phase 2 study had a pre-existing kidney-related autoimmune disease that led to their kidney failure. And these disorders, such as FSGS or IgA nephropathy, frequently recur and attack the new kidney post-transplant. We were able to tolerize 7 of those 10 patients to their donated kidney, and in none of those 7 patients was there recurrence of the pre-existing kidney-related autoimmune disease observed. By contrast, 2 of the 3 patients that we were not able to tolerize to their donated kidney did experience recurrence. And we see this as an interesting initial clinical proof of concept of the extensibility of our approach, not only to induce tolerance to the donated organ, but potentially also to restore tolerance to self. One of the other important observations from our phase 2 study was that we were able to create a stem cell transplant using both CD34 hematopoietic stem cells, as well as our other 2 critical ingredients, facilitating cells and alpha beta T cells. We were able to make these stem cell transplants in patients, notwithstanding profound degrees of HLA mismatch between the donor and the recipient. Ordinarily, you'd need the highest degree of HLA match possible between the donor and the recipient in order to minimize the risk of graft versus host disease. And so on this bar chart, ideally, you'd like a 6 out of 6 or 10 out of 10 HLA match between the donor and the recipient. Almost 3/4 of the patients that we were able to get off all immunosuppression in our phase 2 study, however, had a 3 out of 6 or less HLA match between the donor and the recipient. And notwithstanding those profound degrees of HLA mismatch between donors and recipients, we observed very high rates of engraftment, and we only observed 2 cases, i.e. 5% rate of graft versus host disease, both of which had a common nexus, a known risk factor for graft versus host disease, which led to us to modify the inclusion-exclusion criteria in our study. And since then, we did not observe any further cases of graft versus host disease in the phase 2 study. We've also followed the entire phase 2 cohort, and we've been able to observe well-preserved kidney function in these patients over the course of up to 5 years. The line graph on the left shows the kidney function over time, with the orange line being the 26 patients who came off all immunosuppression, and the blue line being the full intent to treat cohort. And as you see, we've observed very well-maintained kidney function over time. By contrast, in a retrospective analysis of a concurrent cohort of patients who would have been eligible for our study, but did not receive our study, but were transplanted at the same site in the same time frame, our investigator observed declining kidney function over time, which is consistent with what's known of the literature. On the right-hand side, you see a bar chart which shows the rate of reliance on cardiovascular medications of the patients that we got off all immunosuppression, which compares very favorably to what was observed in the retrospective standard of care cohort assembled by our investigator. We'll continue to follow these patients, and we expect to have a median of over 10 years of follow-up on these and other important clinical sequelae for the entire phase 2 cohort at the time of BLA filing. The other important thing that I noted is that we've identified a surrogate marker, which is highly predictive of our ability to get people off immunosuppression and allow them to stay off immunosuppression. Recall that the goal of our therapy is to enable a patient to receive stem cells from a donor and essentially to grow a donor-derived immune system in that recipient, which sees the organ as self. It's very easy to measure whether we've achieved this. We simply look at what % of the patient's immune cells have the donor's genome. And to the extent that a meaningful % of those immune cells have the donor's genome, and they're durably present, we talk about having achieved durable chimerism. And this line graph shows the level of chimerism achieved in the 26 patients that we got off all immunosuppression. And what you see is both T cell and whole blood chimerism levels of north of 90% were achieved in a very high percentage of our patients. And that even at month 3 post-transplant, if patients had high degrees of chimerism, this was highly predictive of their ability to come off immunosuppression. By month 6, all but one of those patients ultimately came off all immunosuppression. And by month 12, this was 100% predictive of our ability to come off immunosuppression. And in fact, it's what the clinician looks to make the determination whether immunosuppression can be safely removed. We've also noted that chimerism, once present, was durable in all of our patients and synonymous with continued tolerance to the organ. And we'll be able to report on 3, 6, and 12-month chimerism in our phase 3 patients at our first and ongoing clinical updates. Our phase 3 registration study, which is currently underway, is an open label study in 120 kidney transplant recipients, 80 of which will receive our therapy and kidney transplant, 40 of which will just receive a kidney transplant. It's almost an identical protocol to our optimized phase 2. And our primary endpoint is what % of the 80 patients receiving our therapy are free from chronic immunosuppression without having experienced a rejection event 2 years post their transplant, which would be 1 year after being weaned off immunosuppression per protocol. We're targeting a total of 18 to 20 sites in the US, and as at the end of August, we had 14 active sites. We'll be providing our first update on a modest number of patients that we've been able to enroll since initiating the study during COVID this November. Overall, what I hope I've been able to communicate to you is not only that we've been able to generate compelling clinical proof of concept in our lead indication, living donor kidney transplant, but we also see the possibility, using the same underlying biology, to extend this basic approach, the same therapy made in the same way and applied in the same way, to a number of additional indications spanning both autoimmune indications as well as other areas where allogeneic stem cell transplant has already been shown biologically to be successful. We'll be rolling out a number of successive clinical trials to demonstrate the applicability and extensibility of our approach in each of those areas of great unmet need. And we see, therefore, the opportunity to create a successive series of franchises, all of which leverage the same product made in the same way, administered in the same way. And that's an important point that I want to emphasize. We do all of the manufacturing in-house. The product itself and the process is less than 24 hours. We don't do any genetic engineering or ex vivo expansion of the cells. And because we do all of this in-house, we control every aspect of the learning, and we haven't had to change any aspect of the manufacturing processes or controls that were material between phase 2 and phase 3. And we're using the same process across all of the indications that we plan to address, other than the deceased donor indication. We have a very experienced leadership and board, and I'm delighted to say that overall, we think that we're very well positioned on the basis of the compelling clinical data that we have to date, our open label phase 3, the 2 additional phase 2s that we'll be kicking off, and 1 more indication that we'll be announcing all before the end of this year, to be able to demonstrate to investors the extensibility and the potential of our approach in a number of areas of great unmet need affecting immune tolerance. We do all the manufacturing, as I mentioned, in-house, and as a result of that, we have significant barriers to entry, not only through the know-how, but the fact that we have a 10-year head start on clinical data of any other group that's been able to show that they can demonstrate reproducible, robust immune tolerance in organ transplant recipients. We're well-financed to success, and we're looking forward to an exciting year ahead. Thank you so much for listening to the Talaris story.